Welcome back to G-Fun Facts Online, the show that dives deep into the most amazing facts our universe has to offer. Today, we're talking about something incredibly close to all of us. In fact, it's in all of us. Our brain, but not just any brain fact. We're exploring its almost magical ability to change, to rewire itself. It's called neuroplasticity, and it's how we learn, remember, and even heal. So what exactly is neuroplasticity? Think of your brain not as a fixed, unchangeable computer, but more like a super advanced, living sculpture that's constantly being reshaped. It's the brain's power to reorganize its own structure, its functions, even its connections, all in response to what's happening inside you or in the world around you. This isn't just about tiny tweaks. We're talking changes from the microscopic connections between single brain cells, called synapses, all the way up to large-scale remapping of entire brain areas. And trust me, understanding this isn't just for scientists in labs. It's the key to unlocking treatments for a whole range of conditions, from brain injuries to mental health challenges. Now, this amazing ability, neuroplasticity, isn't just one single thing. It shows up in a couple of main forms. Let's break them down. First up, we have synaptic plasticity. This is all about the synapses, those crucial junctions where neurons or brain cells talk to each other. Synaptic plasticity is their ability to get stronger or weaker over time, depending on how active they are. This is absolutely fundamental for learning and memory. Two big players here are long-term potentiation, or LTP, and long-term depression, or limited. LTP is like forging a super efficient communication highway between two neurons that fire together a lot. It makes their connection stronger and lasts a long time. Think of it as your brain saying, "Hey, this pathway is important, let's boost it." This is crucial for making and storing memories. Then there's limited, long-term depression. It's the flip side. It's a long-lasting decrease asterisk in how strong that synaptic connection is. This isn't a bad thing. It's essential for pruning away connections we don't need anymore, refining our neural circuits, and even clearing out old, faded memories. It's like brain housekeeping. Next, there's structural plasticity. This is where things get really physical. We're talking actual changes to the brain's structure. This can mean new synapses forming, that's synaptogenesis, or old ones being eliminated, called synaptic pruning. It can also mean the growth of new dendrites, those branchy bits on neurons that receive signals, and even, in some special brain regions, the birth of entirely new neurons, a process called neurogenesis. So while synaptic plasticity is about the strength of existing connections, structural plasticity is about the actual done rewiring and asterisk rebuilding of the brain's hardware. These big structural changes often follow and lock in the functional synaptic changes, creating a more stable foundation for long-term adaptations. It's vital for learning, memory, and especially for recovery after brain injuries. And briefly, there's also something called functional plasticity. This is the brain's incredible talent for moving functions from a damaged area to healthy, undamaged parts. It's like your brain has backup systems and can reroute traffic when there's a roadblock. This often involves a mix of both synaptic and structural changes. So how does the brain pull off all these amazing changes? It uses a complex system of chemical messengers, each with its own job, operating through distinct pathways to get very specific things done. Let's start with the heavy lifters. Glutamate and GABA. These are the brain's primary go and stop signals. Glutamate is the main excitatory neurotransmitter. Think of it as the accelerator. It plays a massive role in that long-term potentiation we talked about, especially by activating special receptors on neurons called NMDA and AMPA receptors. When these NMDA receptors get a strong or repeated signal, they open the floodgates for calcium to rush into the neuron. This calcium influx kicks off a whole chain reaction inside the cell that strengthens the synapse. For example, by adding more AMPA receptors, making the neuron super responsive to future glutamate. On the other side, we have GABA, which is the main inhibitory neurotransmitter. It's the brake pedal, counterbalancing glutamate's excitement. This prevents our brain activity from going haywire and helps refine neural signals. And guess what? GABAergic inhibition, the braking system, is also plastic. The strength of these inhibitory connections can change, and that's critical for shaping how our brain networks operate and learn. The dance between glutamate's go and GABA's stop is crucial for a healthy brain. If this balance gets disrupted, it's often linked to neurological and psychiatric disorders. And get this, recent research even suggests some neurons can release both glutamate and GABA, and they can control the release of each one independently. This adds another layer of incredible precision to how our brain circuits are fine-tuned. Beyond these primary messengers, we have neuromodulators. These are like the orchestra conductors, tuning the process of synaptic plasticity. We're talking about chemicals like dopamine, serotonin, acetylcholine, and norepinephrine. Unlike glutamate and GABA, which have quick, direct effects, neuromodulators have more widespread, often longer-lasting influences. They can change the conditions under which plasticity happens, like altering the threshold for inducing LTP or limited, or how long those synaptic changes last. This allows the brain to adjust its learning mechanisms based on what's going on. Are you paying attention? Are you motivated? The context matters. Then there are the neurotrophic factors, the growth promoters of the brain. These are proteins that support the growth, survival, and development of neurons. A real superstar here is brain-derived neurotrophic factor, or BDNF. BDNF is often released when neurons are active, and it's a powerhouse for plasticity. It helps create new synapses, grow dendrites, enhances synaptic communication, facilitates LTP, helps manage those glutamate receptors, and even supports the birth of new neurons in areas like the hippocampus, which is key for memory. BDNF works by activating specific receptors, mainly one called TRKB, which then triggers pathways inside the cell, leading to changes in gene expression and protein building, ultimately reshaping neurons. When BDNF signaling goes wrong, it's often implicated in conditions like depression and neurodegenerative diseases. Okay, so the brain changes. But it doesn't just change randomly, it does so with incredible precision. This is achieved by activating distinct internal signaling pathways, triggered by different messengers and different patterns of activity. Two fundamental principles here are Hebbian plasticity and homeostatic plasticity. You might have heard the phrase, neurons that fire together wire together. That's a great summary of Hebbian plasticity. It describes how, when a presynaptic neuron, the sender, and a postsynaptic neuron, the receiver, are active at the same time, their connection gets stronger. That's LTP. If their activity isn't correlated, the connection can weaken. That's limited. This type of plasticity is specific to the inputs being used and is thought to be a core mechanism for learning and forming memories. But what if this just kept going? Strong connections would get stronger and stronger, and the brain could become overexcited and unstable. That's where homeostatic plasticity comes in. This is a set of mechanisms that helps neurons and networks keep their activity levels stable, despite all those Hebbian changes. Think of it as a thermostat for your brain activity. For example, a neuron might adjust the strength of all its synapses up or down to maintain a target firing rate. This acts like a negative feedback system, keeping everything in a healthy, functional range. These processes usually work over longer timescales than Hebbian plasticity. What's fascinating is that while Hebbian and homeostatic plasticity might share some molecular pathways, they can also operate through their own distinct mechanisms. This allows for both quick, input-specific learning and slower, overall stabilization. And there's even evidence for rapid, local forms of homeostatic plasticity, kind of blurring the lines between these two. Now, for a really mind-blowing recent discovery, something that just came out in June 2025. Scientists have found evidence that the brain might use separate synaptic transmission sites for spontaneous signals, those that just happen randomly, and evoked signals, the ones triggered by actual experiences. Researchers looking at the visual cortex of mice found these two types of signals have different developmental schedules and rules. Evoked signals, linked to what the mice were seeing, kept getting stronger with experience. But spontaneous signals leveled off. And get this, if they activated normally silent receptors, it boosted the spontaneous activity without affecting the evoked signals. This strongly suggests they're operating at different locations. Why is this so cool? This separation could allow the brain to maintain stable background activity through those spontaneous signals, while at the same time, refining the pathways important for behavior using the evoked activity. It's like having two systems working in parallel, one for overall stability, homeostatic plasticity, and one for experience-based learning, Hebbian plasticity. This gives us a much deeper understanding of how the brain balances being flexible with being stable. And if these distinct signaling modes go wrong, it could be involved in conditions like autism, Alzheimer's, or substance use disorders. Of course, all this change relies on intricate molecular cascades happening inside the neurons. Key players include receptor trafficking. The number and type of neurotransmitter receptors, especially those AMPA receptors we mentioned for glutamate, can be changed rapidly. LTP often means inserting more AMPA receptors into the neuron's membrane, making it more sensitive. Limited can involve removing them. Then there are second messengers. Calcium is a huge one here. How much calcium flows in, when, and where can decide whether LTP or limited happens. Other second messengers like cyclic AMP also play big roles. These second messengers then activate protein kinases and phosphatases. These are enzymes that switch other proteins on or off by adding or removing phosphate groups. They target receptors and structural proteins, changing how they work and contributing to making synapses stronger or weaker. CA, MKUI, and PKA are famous kinases involved in LTP. And for changes that really stick around, like long-lasting LTP and limited, we need gene expression and protein synthesis. The signaling pathways can reach all the way to the neuron's nucleus, turning on genes that lead to the production of new proteins. These proteins then create more permanent structural and functional changes at the synapses. And if that wasn't complex enough, there's metaplasticity. This is like the plasticity of plasticity. Basically, the past activity of a synapse can change how it responds to future attempts to induce plasticity. So, what happened before can make it easier or harder to get LTP or limited next time. Metaplasticity helps keep synaptic strengths in a useful range and stops them from becoming maxed out or completely silent. Those neuromodulators we talked about are key players in metaplasticity. And exciting new research is even looking at how things like transcranial-focused ultrasound might induce metaplastic effects, potentially making brain stimulation therapies more effective. It's crucial to remember that these different forms and mechanisms of neuroplasticity don't work in isolation. They're in constant conversation. For example, functional changes often pave the way for structural changes. LTP might first strengthen an existing synapse, but if that activity is sustained, it can trigger the growth of new dendritic spines or even new synaptic contacts to make those changes more permanent. Hebbian and homeostatic mechanisms are always interacting. Hebbian learning drives the specific changes based on experience, while homeostatic processes make sure the whole network stays stable and doesn't go off the rails. That recent discovery about separate sites for spontaneous and evoked signals really highlights how these processes can be both distinct and complementary. And, of course, experience-dependent plasticity fine-tunes our brain circuits throughout our entire lives. While our brains are super plastic when we're young, we absolutely retain the ability to change in response to new experiences, learning, and even injury well into adulthood. The exact mechanisms might shift a bit, with adult plasticity often needing more focused attention and engagement, but the capacity is there. So why is understanding all these distinct messengers and pathways so important? Because it opens up incredible possibilities for therapeutic interventions. Think about neurorehabilitation. Therapies for people recovering from strokes, traumatic brain injuries, or other neurological damage are increasingly designed to tap into the brain's plastic potential to help them regain function. This includes physical therapy, cognitive training, and cutting-edge approaches like non-invasive brain stimulation, using things like TMS or TDCS, and even virtual reality. Then there's mental health. Many psychiatric conditions, like depression and anxiety, are linked to what we call maladaptive plasticity. Basically, plasticity gone wrong. Treatments, whether they're medications or psychotherapies, might work, at least in part, by encouraging beneficial plastic changes or correcting those unhelpful ones. Remember BDNF? It's a key target in understanding and treating depression. What about learning and memory enhancement? By understanding the molecular nuts and bolts of LTP and limited, we could develop strategies to boost cognitive function and maybe even slow down age-related cognitive decline. And for treating neurodegenerative diseases like Alzheimer's and Parkinson's, which involve massive loss of neurons and connections, leveraging plasticity pathways is a huge area of research. Scientists are looking at therapies aimed at promoting the birth of new neurons, enhancing synaptic function, or protecting existing neurons. That new discovery about spontaneous and evoked transmissions using different sites? That could lead to even more targeted therapies. If a specific disorder is linked to a problem in one type of signaling but not the other, treatments could be tailored much more precisely. The brain's capacity to change, its neuroplasticity, is truly one of its most profound and essential features. This intricate network of distinct chemical messengers and precisely regulated pathways allows for an astonishing level of adaptability. It's what enables us to learn new skills, form lasting memories, and recover from setbacks. From the lightning-fast strengthening and weakening of individual synapses to the slower, more permanent rewiring of our neural circuits, every mechanism plays a vital role. As science continues to unravel the incredible complexities of these separate but interconnected pathways, we get closer not only to understanding the full scope of our brain's dynamism, but also to developing truly novel strategies to promote brain health and treat a vast range of neurological and psychiatric disorders. The journey into the brain's distinct messengers and its incredible plasticity is a journey into the very essence of what makes us adaptable, resilient, and capable of lifelong learning and change. It's a reminder that the amazing 3-pound universe inside our heads is constantly evolving. Thanks for tuning in to G Fun Facts Online. We hope you learned something mind-blowing today. Join us next time for more fascinating facts. Until then, stay curious.